ये कैमरा खुला नहीं कुछ नहीं चल क्या होता है बैठ जाओ ठीक है कंट्रोल करना कंट्रोल भी करना आएगा
So hey everyone, so this is the first time Bioclub and NAC is organizing a talk in collaboration, and we have Pratik Kalel from SBS Integrated MSc Batch 23. Uh, he belongs to SBS, but I've seen from first year he has a great interest in this astronomy, and he is going to give a presentation on one of the interesting questions which humanity has, that how did life begin and could it exist elsewhere in the universe. So the talk of the title is Cosmic Dust to Living Cell, a brief of astrobiology. So Pratik, thank you everyone. So today I am going to give a talk on astrobiology, which is kind of like different kind of subject, interdisciplinary subject, and it is one of the most interesting subjects. Like, but not all people know about it. So I will start with the cosmic dust and try to go to the living sense. So we all are here doing an experiment. We are trying to use various techniques and all and try to create a cell from a cosmic dust. So let's start. Before starting, what is astrobiology? So astrobiology seems like astronomy plus biology. Na? So astronomy, we all know the study of everything in the universe, and biology is the study of life. So combining both of them, we get an astrobiology, which is an interdisciplinary scientific study of life, universe, which includes the origin, evolution, distribution, and future. So astrobiology mainly has two two fields. Like currently, we don't know about the future, how life, like we didn't find life until now in another planets or another universe. So as for now, people are trying to study life on our Earth and trying to implement the same things on other planets and other universe and all. So the two main areas of research in astrology are origins of life and traces of life in universe. So first thing is like find the how we came, like what was the origins of life. Even if we say that there is a chemical, there are various chemical reactions happens and all them combined together something happen and it creates a cell. But still we don't know what was the exact process, and we don't even know, like we don't even have a proper proofs and all. And another area of research was traces of life in universe. We try to find is there any life in other planets and other areas, but still we are not able to find it. So I am mainly focusing on the origins of life. So the content of my presentation will be, I will start with Earth formation. So the main thing which is life, for a planet which should have a life, it should have some basic things on it. So Earth's formation is also one of the important things which we need to study before we start for the how life works. So then we will move toward the compartmentalization, metabolism, and genetic materials. There are some basic um, activities or activities happening in our cell. Like they should have a proper compartments, they should have a metabolism, and they also have a genetic materials. Then we will move forward to create our first living cell, and then we like conclude. So let's start with the Earth's formation. So, as you all know, around 4.5, 4.6 billion years ago, our Earth was not there. Like, there was a sun and there was a dust, a disk of dust, a cosmic dust, we can say, was revolving around the sun. And these dust particles came together and formed a planet of our solar system. So, like that, our Earth was also formed around 4.5 billion years ago. Our Earth was formed and it was looking like this is a small planet. It was not even at the same density which today Earth has. And we will study about its atmosphere first. So the primary atmosphere of the Earth at that time was mainly composed of hydrogen and helium. Like at that time, the first elements which were present in the dust are the most abundant elements. Obviously, they will be the part of the atmosphere. And there was also some little amounts of methane, ammonia, water vapors, which was came because of the volcanic eruptions and all. But this atmosphere couldn't last for long. This atmosphere just flew away just because of the solar flares and solar winds, and Earth isn't uh, like able to like maintain its atmosphere because at that time Earth doesn't have a strong gravitational field. So all of the atmosphere was flew away. Like even today we can see the Mars. Mars doesn't have the enough atmosphere. So after that, one of the major event occurs was the impact of Theia. Theia was another planet which was struck on our Earth around 4.5 billion years ago, which resulted into the formation of our own moon. And also it also gives some amount of mass to our Earth. And this event literally shattered the core of the Earth. So because of this event, what happened? Uh, every rock and all the things was like it was molten, and whatever the elements were present on the Earth, like the denser elements move towards the core, and the lighter elements stay on the surface because of the gravitational field. So, like iron is a dense element. So majority of our core was made up of iron, just because it is dense and it moves to the center. So after this collision, there was an atmosphere which formed as secondary atmosphere. This atmosphere doesn't flew away because Earth already has its own gravitational field, and it doesn't like because of it atmosphere stayed. But this atmosphere was mainly composed of the volcanic acids, which was emitted by volcanoes, and these volcanic gases are mainly reducing, like they were mainly reducing gases, but there are some other gases which were oxidizing. But still, we don't know like whether that atmosphere was reducing or oxidizing. So you may ask why we are like focusing on this, like why we are asking why it should be reducing, why it should be oxidizing. So the major like before moving to that question, what what makes it reducing or what makes it oxidizing? So the once again I came come to the iron. So iron was the compound which was unoxidizing, like it oxidizes. So the amount of iron present on Earth's surface will determine whether the atmosphere will be oxidizing or reducing. If the amount of iron is very large or very much, then it will basically oxidizes and takes all the oxygen present in the atmosphere into it, and it will make it reducing. If its amount was less, then it will not take all the oxygen, and eventually we will get some neutral kind of atmosphere or maybe little bit oxidation. So after that, like, even though after if we see our Earth position, we say that it is in our Goldilocks zone. So Goldilocks zone is like a area where water can be in a liquid form. But if we see our Earth's orbit clearly, it is not exactly in the middle of the old ocean. It is on the inner surface, on the inner edge of the old ocean. Because of it, most of the water on the Earth was just vaporized and fuel. So at that time, Earth doesn't have enough water. But after some time, when volcanic gases was came, they tried to create a greenhouse effect, and also later there was a big event was happened, a late heavy bombardment, where the lots of asteroid was came on our solar system and collided with lots of like all the planets and all. And these asteroids brings water. Even if today, if we see the age of our water present on Earth is more than the age of our Earth itself. So we got an water. If we get a proper gravitational field, we have a reducing environment. I assume it should be a reducing environment. Why? Because if it's not a reducing environment, then we can't produce a life. Because oxygen basically reacts and breaks down all the molecules which are important for the life. So it should be a reducing, but we don't know exactly how much it was reducing. So we had a perfect environment for the uh, for for life formation or for the formation of basic life materials. Now, before moving forward, I would like to say about Miller-Urey's experiment, where he just used some H2O, CH2, H2 gases and in a in an enclosed area, and where it used an electric spark and water was flowing through it. And after some months or some days, he was succeeding into forming some amino acids from his basic molecules. But so this experiment was the first experiment, like I can say the first experiment of astrobiology, where he tried to find how all the molecules could have met. But there was an issue because whatever the environment he has created was strongly reducing atmosphere, strongly reducing. But when we see our Earth's like Earth's history, or when we see all the gases present at that time, we can't say it is strongly reducing. It may ranges from neutral to reducing, or maybe some between. So it's like a spectrum, like a big spectrum. In between, it was belonging, but it was not totally strongly reducing. So whoever is the astrobiologist, they always criticize this. If you are not as astrologer, you will say, "Oh wow, how good experiment this was." But the people who work in this field, they said, "No, this experiment doesn't make a sense." But at least he started the astrobiology, so it is an important thing. So, so after around 3.5 million years, we got our first cell, which was the last universal common existence, Yuga. So that cell has its own boundary, it has its own genetic material, and it has like it can create like it can have multiple reactions following it, and we can say that life was started from this point. But the origins of life start way before promise, like from around 4.5 billion ago to this time, life could like whatever the research is done is called the origins of. They started like how the cell was formed. Evolution started after. Before cell, what was it? And here you can see that like people say that it was started from one point, but according to me, like it is not like that. It was like a bottleneck effect. I will tell you later like why I am saying that it was a bottleneck. So it has like I think there was a several ancestral communities of primitive cells who started the life, not a single one. So we will move forward to the origins of life. So origins of life, we, as I said before, we have to study compartmentalization, metabolism, and genetic materials. A cell, to be said a cell, it should have these three. So let's start with compartmentalization. If we see our modern cell, its cell wall or cell membrane looks like this. So it has a phospholipid bilayer, some cholesterol proteins, there are some cytoskeleton filaments, and also some carbohydrates and all. So this is like a complex cell membrane. But when we think about like how the first cell membrane could have been formed, we can't start with this because it is very complex and it's very hard to think. So we can start with a very small molecule. Like here you can see there is some like a phospholipid bilayer. We can take those phospholipids, but very small or a small molecule. So let's take amphiphiles. Why amphiphiles are the best candidates? So they have an ability, like at a particular pH and temperature, they can create a vesicle. So vesicle is like a bilayer, cylindrical bilayer, in which they separate out the environment, where you can concentrate the materials and start a uh, life supporting reactions and all. So these amphiphiles has an hydrophilic head and a hydrophobic tail. Hydrophobic tail is around 8 to 18 carbon. If it's less than 8 or it's more than 18, we couldn't form vesicles. So we need to take this kind of this kind of amphiphic molecules. So these amphiphic molecules form all of these structures, but we are mainly focusing on the vesicle. Other structures also are best, but they don't form any another like a different environment or encapsulate an area where we can form. So how the vesicles form? So every molecule has a pK value. pK value is like how much the total molecules are protonated or deprotonated. Like if I say I had some 100 amplifiers and 50% of them are protonated and 50% of them are deprotonated. And when these scenarios are happening at a particular pH, like if I say at pH 7, they are they have 50% protonated, 50% deprotonated. Then I can say that it is its pK value. So like different kinds of amplifiers have different kinds of pK values, and these pK values determine whether they will form a vesicle or not. So let's move forward. Let's take one amplifier. Can only one amplifier be enough? What do you think? If we see here, if you take only one amplifier, let's take like a random amplifier with a square head and a long head, or a triangular head or long head. And I just try to bring them together and at a particular pK value, they will form a vesicle. So when they make a vesicle, if I see their outer boundary, they are tightly packed. So not a single molecule can enter inside them. So is it good? If we see today's cells, they are semi-permeable. We need to have some molecules like there should be some molecules enter inside the cell so that accumulation of the molecules could happen and they can further uh, start some reactions and all. So we can't use only one only one amplifier. What if I take two amplifiers? What will happen? So if I take two amplifiers, they will have some gaps in between. So these gaps will uh, allow us to enter molecules from outside place. But if I take more amplifiers, like more number of amplifiers, I try to make it more complex. Will it be better or not? What do you think? At a certain point, like uh, if I increase complexity, at a certain point, it will be as more permeable. Like we don't want a more permeable and we don't want impermeable. We want in between them, semi-permeable. So it can't be good. Like it is not good. So we need a certain amount of amplifiers which can come together and form a vesicle, and it should be a semi-permeable. So there was an experiment which my lab has done. So they took three molecules. One was like they are L1 carbon chain molecules. One is had with acid group. Another one was made up of glycerol, and the third one was made up of alcohol. The head groups are made up of it. And what I did, I first tried try to form vesicles only using acidic head. When I see it, like they can form vesicles only at pH 7 and pH. When I try to combine acidic group and glycerol group, their range increases. Like you can clearly see the vesicle number was increased, the vesicles become little bit bigger, and they ranges from pH 7 to pH 8. What if I do acidic group and OH group? If I use both of them, they also increase the range from pH 7 to pH 11. But you can see the number of vesicles present here are very less as compared to the P1. It is maybe because of the OH interaction between OH group and the acidic group. So in the third one, I use three of them and try to create a vesicle. And it is one of the most good one, because you can see it was start from pH 7 to pH 11. It can completely form a vesicle. So the more you make a thing complex, it will definitely um, uh, survive for a longer period. So we talk about the vesicles. Are the vesicles are only key for compartments? Is there any other thing which we can do? What do you think? Hmm? There is another thing named as conservates. So these conservates are the droplets which are formed by the separation of polymers and charged molecules in water. So you can clearly see here there was a protein, a polysaccharide. They both combine and form conservates. So these conservates basically concentrate the molecules. So whenever they are present, they literally absorb all the elements present around them. And the first thing which we need, like the to happen a reaction, we need to concentrate the molecules. So these people do some of the research, like research also has done in this field, but like not that much. So these conservates can grow much divide under a physical changes, like crossing through a rock pores and all. So now we move forward to metabolism because we have created the physical. Now, what is metabolism? Metabolism is the sum of all the reactions occurring in our body. But when we talk about prebiotic conditions, there was no body, not a single body. So what are we are here going to do? So we will basically try to mimic all the pathways happening in our body, whether they can be done without using enzymes, because enzymes came way later. So for a metabolism or to form a life, the main component is carbon. Carbon is an tetravalent element. It has a catenation. It can form a large branches, a uh, large chains and all, which other molecules couldn't able to touch, couldn't able to. Also, their bonds are strong, so they can be used for a structure to form structure. And also, they were the most abundant material present. So obviously, we will use it. So, what we are looking into a metabolism? Is it like whatever the metabolism you are looking for? Is it resembles to the modern day metabolism, or it has like some different kind of metabolism which we are going to? Also, when we see enzymes, what do enzymes do? Like they basically speed up the reactions. If I don't use enzyme, reaction will happen, but it will take time. It will may take some days or some months and all to complete a reaction. So enzymes is like a work. So when we see the metabolism, it goes to a catabolism with the oxidative degradation, and it forms some common currencies which we can use for the anabolism. So we first have to find a fuel source. Then through the catabolism we can convert into the common currencies, so that we don't like we didn't need to rely on a single fuel source and all. So they will just take the fuel source from any place and they can just store like a battery, like Na plus gradient, it depend H plus gradient, and they will further can do a anabolism. So prebiotic analogs of anabolism. How could the anabolism could have happened at that time? So the first thing, like we can say, like larger molecules could have been formed from the smaller ones. So at that time, CO2 was most abundant, and metals was present on the Earth's surface. So from the CO2 and metal ions, we can form methanol, formate, and methane-like structures, which we can use further for the other molecules to make. Also, if we see a carbon monoxide in the presence of UV light and sulfide, it forms a glycolate. And if I combine glycolate with sulfide and do the glycolate irradiation, I can create citric acid cycle. Here you can see, like I didn't use a single element. We can create citric acid components. And there was another molecule named cyanide, which was one of the most abundant at that. And using a cyanide, some basic metal ions, and a UV light, we can create half of the amino acids, all the amino acids. We can also create pyrimidine, purine, ribonucleotides, also some precursors to lipids. So whatever the reactions here happening are completely independent of the modern biology. Like they are different. They are just trying to mimic. So let's move to the catabolism. So glucose 6 phosphate with iron and high temperature, we can create a pyruvate. It will take time. Like, if we see the glycolysis process, they use a bunch of enzymes to do that reaction in very less time. But we can do this reaction, and it will take some time, but it will happen. Using a pyruvate and glycolate, same citric acid molecules can be formed. Using oxaloacetate, we can move towards the energy analogs, the energies like ATP and all. And also from oxaloacetate, using a hydroxylamine and iron, we can further go for the amino acids. So whatever the things are happening here, you can see like what are the elements which I have used were present at that time. And you may say why, how you know that where they are present or not. So some research has been done. I couldn't cover all the things here. I have added some references. You can take a look. Further, you can I can say glyceraldehyde and glycoldehyde in the presence of like before I use a metal ions in the last reaction. Majorly, I have used a metal ions. And what if I wanted to create a same thing without using metal ions? So using a cyanide and diamine phosphate, I can create a pyruvate or phosphinol pyruvate, which further can create all the things if we want. And also a mixture of pyruvate, glycolate, and H2O. They can start some cycles like 4-hydroxy to keto cycle and malonate cycle, which are one of the important cycles in our pathways. And these cycles leads to the aspartate formation, also other amino acids, and also they work in the way so that carbon cannot be like all the carbon molecules should be used. So and basically they balance the carbon and energy. So that's how all the reactions we can create without we can. And another one major thing I want to tell you that was wet and dry cycle. So when we see in our labs, like we try to do a, so it's like a we are trying to create some molecules from physical. The same thing happens in on Earth. So here we can there is a pond. There is a pond. There is a sun, and if sun was so much harsh, it will basically evaporate all the water present in the pond. So in the pond there are various like there are clay. In the clay there are various metal ions. There are all other molecules are there. And when we do this like if sun evaporates, it will concentrate. And after that it rains. We say it raining. If it's raining, the pond will fill again. So it will again become wet. So it lead to a dehydration reactions. So these dehydration reactions are one of the most important things, because if we, like say, if I wanted to create a big molecule, I will first try to concentrate the smaller molecules so that they will bond to each other and they will create some complex molecules. After that, uh, I get to a bedside. So whatever the molecules from this molecule, whatever the bigger molecules are formed, the stable ones will remain same even after it becomes big. But the which are not stable, they will again become a smaller, like the small molecules. And this cycle I did repeatedly, and they actually form a lot of molecules using only wet and dry cycles. And these stable molecules can further uh, contribute into a molecular evolution. So now we see like how we can form all these molecules. And the main thing which we get to know is that to concentrate the smaller molecules. So to concentrate it, at that time, the two methods we can use. The first thing is hydrothermal vents. So hydrothermal vents emits like hot gases and all. Because of it, the water around it becomes hotter, and it start to flow from the ocean, and it creates some barrier. Like if water is warm, it has some salt concentration in it. It will definitely create a barrier. It's not exactly a barrier, but an environment or area where materials can be concentrated. So using it, we can concentrate the materials, and you can see that some pathways are trying to form. Another thing is using vesicles. I made a vesicle, and somehow we try to concentrate the molecules, and it will do the same. But while concentrating or uh, like making a barrier has some limitations. Like once we make a barrier, like not a single carbon can go outside, not a single carbon can go. So it will say that we has a carbon deficiency. Like we have a limited amount of carbon which we have to use for different pathways and different cycles. So research, like uh, for some researchers say that if these uh, compartments or these concentrated places manage to form a nucleotide, nucleotide gives a positive feedback for a metabolic activity. Another side, amino acids also support this. But on the other side, lipids don't. So we also have to take care about the carbon. Like it should be used for all the pathways. It shouldn't be like only one pathway. And that's all we learned about the metabolism. We start from like from the prebiotic era. Now, what if I say like there is another way of approaching this? It is called top-down approach. So we basically what we do, we see today's life, see today's environment, see today's pathways and all, and try to mimic it or try to go back into it and try to find if there is something behind it. So when we see a ETC, electron transport system, it's basically a proton gradient. It creates a proton gradient where the element proton gradient, and we can say it is a potential bridge between evolution history and protocellular stage, because we can say that hydrothermal vents also can be a ETC, because it's providing various molecules through the gases which it's emitting. Again, it definitely has some proton. So we can say that hydrothermal vents are also causing some proton gradient. So here you can see there is an like on the upper side there is a full acidic to neutral environments. Uh, Going little bit down, we find some hot alkaline environments, and if we go to some places, there is some hot neutral and acidic environments. And every place, like uh, there is an uh, formation of like different molecules and proton donation and taking was happening. So it's not like perfect, but we can say it can be. So hydrothermal vents also has some other things, which is like uh, they have a minerals which can catalyst and start some redox reactions. They can they can generate protons across inorganic barriers and also create a gradient, whatever I said before. So that's how the metabolism should have been worked at that time. Now we create a compartment. We see how the primitive metabolism would have been happening. Now we will see to create a genetic material. In modern biology, we see DNA and RNA. So just for a presentation, I have showed here DNA is a two strand and RNA is one strand, but they can be a double strand or single strand. Uh, so in today's like DNA and RNA was the genetic materials. But what will like which one comes? DNA or RNA? What do you think? RNA. What if I say both of them generated like simultaneously or they evolve at the same time? They can. I will show you some papers. I didn't include that group here. So if we see RNA and a DNA, RNA has capability to react. Like it can easily react because of its reactive hydroxyl group, and DNA. It is like it is more stable than it, and it is more good for storing genetic material, like storing information. So I think RNA must be the primitive genetic material, but some people say DNA also was present. Still, let's go with RNA. So let's build an RNA. How we can form a RNA without using it, and the DNA replicating all over the place. How the first RNA molecule was. So there are two main possibilities for the primitive RNA formation. One is like activated monomers initiate RNA synthesis and they come to the form RNA. Another one is like short oligonucleotides. They form in such a process and they just gather with the ligation and you, with the two-stage assembly they form an RNA. So if we try to build an RNA, we can take some basic elements like whatever I showed here. Using these elements, we can doing all those reactions. We can go to the RNA. I'm not going to explain everything, but there is a catch. Like if you see the ribose sugar, the 3 1, third one, and the 10 1, they don't. They basically have some problem with each other. Like cytosine and ribose have some issues. Like ribose, basically ribose doesn't like mostly unreactive, and cytosine also has a poor metabolism. If I wanted ribose to be reactive, I had to make an acidic environment, and I will try to make it acidic. But at the same time, cytosine's nucleophility will reduce, and because of it, they won't be able to make a bond. Also, the same another problem comes with adenine because it has a multiple tautomers. That the adenine which you wanted, it's only a 0.1% of all the tautomers present. So their bonds cannot be formed. So what we can do for it? Why we are like forming a ribose independently inside? We can structurally use some another thing like uh, structurally we can go by other side. So so to build an RNA, we will start with a glycoldehyde and cenamide. They will react to each other in the presence of phosphate, and they will do some kinds of reaction and make an two amino oxidol. Then I will with the glyceraldehyde and amine of sugar, they will react and they will form some isomers and all. And one of their isomers was this riboamino oxazide. Using this riboamino oxazide, this riboamino oxazide basically crystallizes and gives the arabino isomer. So this arabino isomer further do some reactions and all with the with the molecule cell, and they will form an unhydronucleoside. So this unhydronucleoside gets reacted with the urea, and it basically further goes down to form ribocyclisine 2030 cyclic phosphate. Using this uh, element, you can see there are multiple isomers of the same element. So what will we do? At that time UV was present. So if we take this under a UV, all other isomers will disfigure. And there was one isomer will remain. Where you can see the cysteine will be converted into uric. And that's how we can form a first RNA. So this process is very complex, and whatever this was done and is done under our lab. So we don't know whether this process was happening at that time or not. But we can say this is the like most near thing to that. So we form a RNA monomer, but we need a long chain of monomers, a polymer, RNA polymer. For that, RNA replication should be there should be RNA replication. So in today's world, like in modern biology, we need uh, enzymes like RNA polymerase to polymerize the RNA. So polymerase to replicate the RNA. So it will replicate and make multiple copies. But at that time, these enzymes were not there. So if we see today, like if you wanted to create enzyme is basically a protein. If you wanted to form a protein, you have to go through a central dogma process and then you can have a protein. But if you wanted to create an RNA, you need a protein. If you wanted to create a protein, you need an RNA. So it's a loop. So it's like what came first? If you say RNA came first, then how it polymerizes, how it like big chains of RNA forms without using enzymes. How? You don't know. So people try to like try to find it and say that what if we do non-enzymatic RNA? Like it's been more than 60 years. People are trying to find it, but still they couldn't able to find it. So we don't know how RNA replicates without using enzymes. But there was some other researches was done, like when was this was done? They said like what if RNA itself used as an enzyme or itself as a as an enzyme. So by doing some research, Thanos in 1982 discovered a ribosome. You all know the ribosome is an enzyme made up of RNA. But what ribosome do? It just cut 2 or 3 monomers on one side. It doesn't like replicate a whole molecule. So they try to make it. They try to do a wait and try cycles and all. And they find that until now we only try to make a long RNA chain until 26 months. After that we couldn't able to. So we don't have a proper proof like how exactly the RNA was replicated. So we can we say that then how this would have been happening? So some scientists say that why ribonucleotide? We can use another like why ribose sugar? We can use another thing, like peptide nucleic acid, ribonucleic acid, ribonucleic acid. So these molecules can be created without using, and they also have a strong bonds, they can work better than RNA, but we can't see these molecules today, and we don't know even if the molecules is present at that time or not. But some scientists artificially try to create these molecules, and they're working very good. So maybe these molecules were present at that time. And another thing which I wanted to say, why nucleic acids like AUGC and ATGC? It's like if we see our RNA and DNA, they have these five molecules, and all our living system was based on this. Is there any another molecules present like this? So some of the scientists like try to draw structures and all, like all the possible pyramids and uh, they they create a lot of structures and they find some of these molecules like they can be worked as a nucleic acids. So whatever the molecules which we use in our DNA and RNA are known to be canonical nucleic acids. And when we see these are the non-canonical because they are not used in the modern DNA and RNA, but still they are existing, like they are present, and maybe with using these molecules we can create an RNA. So another interesting thing which I wanted to tell you that these non-canonical nucleic bases or a canonical nucleic bases was found in a meteorites. There are some carbonaceous meteorites named Parkinson and Lonewood, which was taken from the Australia and Antarctica. They when they see this asteroid, they found all of these non-canonical or canonical nucleic bases. When we see, we all know, like, do you all know panspermian theory? It basically say that Earth, like on our Earth, life was not formed. It came from somewhere else. But what if I say these molecules may have come from somewhere else? Because these molecules, like forming these molecules in our prebiotic Earth conditions, very hard. And maybe these methods would have been helped to create a say, like create these molecules and these RNAs and DNA structures. So one thing which I never did in the vesicle thing and RNA, like the vesicles are majorly formed in the alkaline conditions, like ranging from like 8, 9, 10 even pH. Even I showed that diagram where you can see that we start from pH 7. So majorly all the amplifiers create vesicle in a basic condition. And if we see an RNA, it stabilizes in acidic condition. If we make a basic condition, RNA will take. So how these two molecules came together in form a cell? Because we need a compartment made up of vesicle, made up of like we need a compartment of amplifiers, and we need a DNA present inside it of RNA. We need a which is acidic. So they are contradicting with each other. So if I wanted to create a vesicle, I can't form RNA. If I wanted to form RNA, I can't form vesicle. So what could be a solution for this? So one of the recent is that what if like the amplifier has a net schedule? What if I add nucleic base and phosphate on its surface? So it will be a stable structure. It will form a vesicle and it presents some nucleic base inside. So these nucleic bases uh, can further create RNA or DNA inside the vesicle. And another thing, like as I said, like all the amplifiers form vesicles in alkaline conditions. There are some amplifiers which can form in acidic conditions, but they are not so much. So now we know like genetic material somehow we try to create. Uh, we go through the metabolism, we go through the uh, compartmentalization. Let's try to build a cell. Before start to build a cell, I would like to tell about the geothermal cell. So these geothermal springs majorly present on the Earth's surface, and they emit metals, minerals, and all, and they have some clays. Clay has catalytic ions and all of it. So these, like if this clay, like when I was in Pune, they bring this clay and try to form a vesicle in the presence of the clay. So clay was somehow supporting the vesicles to create to sustain for a larger pH level. Also, they can start some basic metabolic processes, and also they can do some other preventive activities. So these geothermal vents also a major thing, like thing with the hydrothermal vents. So let's let's say we have we have a numerous amount of lots of ponds present on Earth, from the high mountains to arid lands towards the ocean. And these ponds are basically distributed in two different types. Like one are acidic, one are basic. So acidic ponds, where we, with the wet and dry cycle, all the metabolic activities, we can form an RNA. So it is like an RNA synthesis cycle. And if we go for the basic conditions, it can basically form vesicles. And suddenly it ends. And a stream was started to flow and try to connect all these ponds and move towards the ocean. So this stream would bring these elements, whatever the RNA was formed, whatever the vesicles formed, will bring them together. And with some metabolic activities, we can have our process. But there should be some RNA or there should be some vesicles which should sustain at a particular pH or at a common pH. At that pH, RNA should also uh, should not degrade, and vesicles also should be in a strong form. So by doing this, we can basically form a cell. Another thing is hydrothermal vents. As I said before, the hot stream of water going through pores of rocks and all. So these rocks will basically have can make some primitive divisions in between the vesicles and all, and these are able to form a current electricity. So that's how we will get our first living cell. So that's how all the processes work together to form our first living cell. And whatever I have did until now in this talk, it was on the basis whatever I have read and all the papers in my internet. And it's not even a tip of iceberg. It's very there are very vast other researches happening and all other uh, like uh, people are trying to find how it can happen. Astrobiology is like a field there you can't just say like this could have been happened, this happened, that happened. No, we are assuming like this could have been happened because these processes have been happening around billions of years ago, and from that now Earth was changed completely. So we don't know how was the conditions present at that time. We are just assuming and try to create our own theories to satisfy ourselves how we can. So I would like to thank all my Sudarajmani and team, full left as a with them also by people. So thank you Pratik for the talk and